Hello, everyone. Welcome to Brain Blitz Audios. In today's episode of NEET Preparation, we'll be looking at previous year questions of biology in medical entrance examinations in India. Today, we'll be dealing with the chapter Molecular Basis of Inheritance. This is the sixth chapter in the grade 12 syllabus of biology in CBSE. Let's continue from where we left off in the last episode by moving on to this first question. During protein synthesis in an organism, at one point, the process comes to a halt. Select the group of the three codons from the following, from which any one of the three could bring about this halt. So we need to find over here the set among the options which contains codons that stop protein synthesis. So the codons which stop protein synthesis are known as stop codons or nonsense codons because they do not code for anything. Now, how do we know which of these are the nonsense codons? Well, let's look at our table, shall we? You see that UAA, UAG, and UGA are given as the three stop codons. So there are only three stop codons out of 64. So and they are UAA, UAG, and UGA. So option C is the right option. If you look at all the others, you find that UUU goes for phenylalanine. However, UCC goes for serine and UAU goes for tyrosine. So that means they are different and they are not stop codons. And UUC is also phenylalanine. That itself proves that this option is incorrect because that's not a stop codon. And UUG is for leucine. Again, not a stop codon, so option D is also incorrect. The right answer is option C, UAG, UGA, and UAA. So these are the three codons which can stop the process of protein synthesis. Let's move on to the next question. The total number of nitrogenous bases in human genome is estimated to be at 3.5 million, 35,000, 35 million, 3.1 billion. We need to find out which of these is the right one. So genome is usually measured in the number of nitrogenous bases. And in terms of numbers, we know that one human genome contains 3.1 into 10 raised to 6. So 3.1 into 10 raised to 6 is the number of chromosomes. Bases in the human genome. And that points to the fact that option D, 3.1 billion, is the right answer. Now, let's look at this question. Which one of the following pairs is correctly matched with regards to the codon and the amino acid coded by it? So we're given a codon and an amino acid. We need to find out which of these codons codes the amino acid given. So again, let's use the help of that table. Option A states that UUA codes for valine. Let's find out UUA. That's right here. However, it codes for leucine. And valin, which is VAL, is coded by GUU, GUC, GUA, GUG. So none of these are similar to UUA. So option A is incorrect. What about option D, CCC alanine? CCC is right here. However, it's in the category of degenerate codons that codes for PRO and not for lysine, which is LYS. So option D is incorrect. Alanine, which is represented as ALA, is coded by GCU, GCC, GCA, and GCG codons. What about option C, AUG cysteine? Now, if you look at cysteine, which is CYS, it is coded by UGU and UGC and not by AUG. AUG is a non-degenerate codon which codes for only one amino acid, and that is methionine. Option C is incorrect. If you look for lysine, which is LYS, you find that AAA and AAG code for lysine. So therefore, option B, AAA codes for lysine, is the right option. Let's move on to the next question. What is the error in the above diagram? Arrows are wrongly depicted, polarity is incorrect, both arrows and polarity are incorrect, none of the above. Now, this diagram given here is for the process of transcription of DNA. So you notice that these are RNA, and this is the parent DNA that is getting transcribed. And if you notice at the RNA, you see that at the bottom end, the RNA goes from the 5' end to the 3' end. And for the lagging strand at the top, it goes from 3' end to 5' end. And there lies the error, because lagging strand is shown from 3' to 5'. However, the RNA polymerase only codes an RNA sequence from a 5' end to a 3' end. So if the DNA sequence is from 3' to 5', well and good, it can form a leading strand. If it's from 5' to 3', then it codes in the opposite direction and it forms lagging strands. So the polarity is incorrect. The polarity of the lagging strands are incorrect. So option B is the right option. Now, if you say that option A, arrows are wrongly depicted, that's incorrect, because any one of the strands can be leading or lagging. And since the arrows are correct, that means that A and C are incorrect. And since we found out that B, polarity, is the reason why this diagram is full of errors, then we can say that option D, none of the above, will also be incorrect, because we found an error. The right diagram will be something like this. So we have 5', 3' for the DNA, and over here it's 3', 5' for DNA. Now let's draw a lagging strand here. And the law is that all RNA strands are from 5' to 3', because that's how RNA polymerase codes. So this end should be 5' and this end should be 3' for RNA. In the above case, it's been inverted. And the leading strand can be as it is, 5', 3'. And this gives you the correct diagram. And with the polarities corrected, the original diagram looks like this. And that's why option B, polarity is incorrect, is the right option, because an RNA polymerase enzyme only codes from 5' to 3' end. So when you're having transcription, all strands, whether leading or lagging, codes from 5' to 3' end. Let's look at this question. TATA box of eukaryotic promoter lies about 25 base pairs, 50 base pairs, 75 base pairs, or 200 base pairs upstream of transcription. Which of these is the right one? So what is a TATA box? Now this TATA is a DNA sequence, which is a promoter that indicates where genetic sequence can be read and decoded. So the reason why it's given TATA box is that the sequence, the DNA sequence, often reads like this: thymine, adenine, thymine, adenine, 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 TA. This is usually the sequence. And this sequence is always found 25 base pairs before the transcription site. And before translates to upstream. So therefore, option A, about 25 base pairs upstream of the transcription start site, is the right option. So anything more than that, there is virtually nothing but normal DNA, but a specific promoter always lies about 25 base pairs upstream of transcription site. So B, C, and D are all incorrect because the specific sequence given here, that is the TATA box, always lies about 25 base pairs upstream, and it is always having the code thymine, adenine, thymine, adenine, adenine, adenine. And this is the DNA sequence that indicates where genetic sequence can be read and decoded. Let's move on to the next question. Which one of the following correctly represents the manner of replication of DNA? So before we check into all the diagrams given here, there are two rules. First rule, new RNA strands are formed in 5' to 3' direction, and they are complementary to DNA polarity. This is number one. Number two, mRNA synthesized, so the two strands of mRNA synthesized are always anti-parallel to each other. So one is the direction of the relative direction of the strands, and two is the polarity of the RNA strand, the mRNA strands. So we need to find out which of these is correct. Let's look at the first diagram. So you see over here, the obvious error is that both mRNA strands are pointing from 3' to 5' end, whereas the first rule states that new mRNA strands are always found in the direction 5' to 3'. So therefore, option A is incorrect. What about option B? So over here, the RNA strand is 5' to 3', agreed. And in B, it goes from 3' and ends at 5'. So that is one obligation that we find. That's one obligatory error because, again, RNA strands are always from 5' to 3'. However, we can also notice that both these strands, when you keep them together, are actually parallel to each other, where in reality, they must be anti-parallel to each other. So the direction of the strands are always anti-parallel. So option B is incorrect. What about option C? Now in option C, you notice that both the DNAs are anti-parallel and they are correct. If you look at the mRNA, you find that the leading strand is from 5' to 3'. However, the lagging strand is from 3' to 5'. Again, a breach in the direction rule. New strands are always formed from 5' end to 3' end. So option C is incorrect. The right answer is option D. Now in option D, you see that both the leading strand and the lagging strand begin at 5' and at 3', and both of them are anti-parallel to each other, and they're also complementary to their respective DNA. So that's why option D, among the given 4 options, is the right diagram. Let's move on to this question. Select the correct option. Now we're given one table with 2 columns. Direction of RNA synthesis is 1 column, and the other column is the direction of reading of the template DNA strand, the original DNA strand. So again, we use the first rule here. The first rule states that new strands of RNA are always from 5' to 3' end, and that they are complementary to the direction of parent DNA. I mean, the template DNA. So since new RNA strands are always from 5' to 3', we can eliminate options B and D, because options B and D say that the direction of RNA synthesis is from 3' to 5', which is not possible. Now, among the two given options, A and C, which have the direction of RNA synthesis as from 5' to 3', let's look at the second column. One of them says 3' to 5', and another one still says 5' to 3'. When you look at our first law, you see that they have to be complementary to the direction of the parent DNA, anti-parallel to each other. The template DNA is anti-parallel to the RNA formed. So therefore, option A will only be the right option. It satisfies both conditions. Condition number 1, direction of RNA synthesis is from 5' to 3', and condition 2, they are complementary to the direction of the parent DNA, so the direction of reading of the parent DNA, the template DNA, has to be opposite, anti-parallel, which is 3' to 5'. And that's why option A is the right option. Next question. Which one of the following represents a palindromic sequence in DNA? Now, what is a palindromic sequence? Now, this is a sequence of genetic material which is read the same when read from both strands. So if you read it from 5' to 3' on one strand, and you start reading it from 5' to 3' on the other strand, the code still stays the same. The code does not change. Now, let's look at option D. The first strand says 5' GATACC. Now, the second strand says GAA. Now, over here, there is a mismatch because the third on the first strand is T, and the third code on the second strand, the third nucleotide on the second strand, is A. Therefore, these are not palindromic. Option D is incorrect. Option C. 5' to 3', the first strand reads CATTAG. However, for the second strand, it reads CAATAG. So in the first strand, there are 2 T's. However, in the second strand, there are 2 A's. Another objection that we can see is that thymine is bonded to thymine. Again, not possible. So option C is incorrect. What about option B? The first strand reads CCAATG. However, the second strand reads CCTAAG. So they're not palindromic. That is reason number 1. And reason number 2 is some of the bonds are not correct. I mean, G bonds with C, but T does not bond with C. A bonds with T, but A does not bond with A. A does not bond with C. So there are a couple of errors that way as well. So option B is incorrect. The right answer is option A, which is GAATTC on both ends. See, GAATTC, GAATTC. G triple bond C, A double bond T, A double bond T, T double bond A, T double bond A, C triple bond G. So they are, so the nitrogenous basis bonding is also correct, and they are read the same on both strands. As we move along both strands, it is read the same way. So option A gives you a representation of a right palindromic sequence. The correct palindromic sequence is found in option A. Now, Let's look at question. Let's look at this question. 30% of the bases in a sample of DNA extracted from eukaryotic cells is adenine. What percentage of cytosine is present in this DNA? How do we find out cytosine when we're given adenine? We use the Chargaff's rules. Chargaff's rules state that rule number 1, number of adenine is equal to number of thymine, and number of guanine is equal to the number of cytosine. So because, A is bonded with T always and G is bonded with C always, so if you know the number of A's, then you must also know the number of T's in the complementary strand. And if you know the number of G's in one strand, you will know the exact number of C's in the other strand. So their number would always be the same. And rule number 2, which says that A plus T, the number of A plus T divided by the number of G plus C. Now this may vary from species to species. This ratio can vary from species to species. However, one thing that is common is that when you're given a percentage, A plus T, and then you're given the sum of the percentages of A plus T and G plus C, that always leads to 100 because these 4 nitrogenous bases make up the whole DNA. So this has to be true. Now, using these Chargaff's rules, let's start solving. So it's given that 30% is the number of adenine bases. So since A is always bonded with T, we also know that the number of T will also be 30%. And so that means number of A plus number of T will give you 60%. And since we know that the total percentages add up to 100, so the number of G plus C can be taken as 100 minus the percentage of A plus T. So 100 minus 60, that gives you 40%. And again, since G is bonded with C always, so the amount of G will be 20%, and the amount of C will also be 20%. So that makes option B the right option. Using Chargaff's rules, we've calculated the amount of thymine, and using the second rule, we found out that the combined the combined percentage of guanine and cytosine in that DNA, and then we split it into 2 to get the percentage value of cytosine present in the DNA. And that value is option B, 20%. Let's look at this question. There are 3 genes, A, B, and C. Percentage of crossing over between A and B is 20%. Percentage of crossing over between B and C is 28%. And percentage of crossing over between A and C is 8%. Give me the sequence of genes on this chromosome. So, what is the correct sequence? Now, if you remember, the percentage of crossing over of B and C is 28%. And when you add the percentage of crossing over of A and B and A and C, you will get 20% plus 0.8%. That is equal to 28%. So the sum of the percentage of crossing over between A and B and A and C will give you the value of the percentage of crossing over of B and C. So basically, if B is crossed over with A, which gives you 20, 20%, and A is crossed over with C, that gives you 8%, then the total crossing over would be 28%. And that's why option A, BAC, is the right answer. If you kept it as ABC, then that is A plus BA, AB, that is 20%, and then BC, that is 28%. So that will be equal to 48, which is not equal to 8, because A and C is given as 8%. So this is not true. Try ACB, for instance. So AC is 8%, BC is 28%. You get 36, you won't get 20. So therefore, that option also fails. And since we found that BAC is the right one, we can also say that option D, none of these, is also incorrect. So just by doing a simple addition, we found the correct sequence by adding the percentages of crossing over between the 3 genes. So that's a simple and easy way in order to solve this problem. So, next question. Which one of the following group of codons is called as degenerate codons? So, degenerate codons are equivalent to stop codons, because these codons do not code for any amino acid. And what are the 3 stop codons? Those are UAA, UAG, and UGA. So therefore, option A gives you the right answer. UAA, UAG, and UGA are the stop codons, which do not code for any amino acid. Stop codons are also known as degenerate codons. Now, the given figure shows the structure of nucleosome with their parts labeled as A, B, and C. We need to find out what is A, B, and C. So the structure of nucleosome, this represents the packaging of DNA. And when you write the process, it is given that DNA is wound around. So it is wound. So this is spun on the edges of a round histone octamer with the ends of the DNA attached to H1 histone. So we see that both ends of the component A are attached to B, and A is wound around C. So in accordance with the statement given here, we can safely conclude that the structure A has to be DNA. DNA is always wound around a circular spherical histone octamer. So option C will be histone octamer. And the lean component B, which holds the 2 ends of DNA, is H1 histone. So that means option A is the right option. If you look at option C and D, these are showing the genetic material as RNA. However, packaging is usually found in DNA inside nucleus. So options C and D are incorrect. Now, if you look at option B, it says that A is the H1 histone. However, histone particles are both histones are negative, and so negative always repels negative, so this will not be possible. And DNA, being such a short strand, will also not contribute to economical packaging. So that's why option A, which shows the structure A, the long coil, as DNA, the short stub that holds the ends as H1 histone, and the sphere as a histone octamer, that option A is the correct structure of a nucleosome, which packages DNA. Let's look at the last question of this episode. Match the codons given in column 1 with their respective amino acids given in column 2 and choose the correct answer. So we're given UUU, GGG, UCU, CCC, AUG, and we have the amino acids serine, methionine, phenylalanine, glycine, proline. So PRO stands for proline. So how to solve this question? Now, usually when we're matching, it's best to find out the easiest. Now, if you notice over here, the only non-degenerate, the only codon which does not have a shared, which only codes for one amino acid is AUG. So AUG only codes for methionine. So therefore, E2 is correct. So E is always matched up with 2. So that eliminates option D. So how do we proceed further? Let's show you that graph that I just did. So this graph will give you, this table shows you all the 64 codons and their connections with the 20 amino acids. So as I said, AUG always codes for methionine. And you can have your proof right here. Now, for the codon UUU, we find that it shares with UUC in order to code for PHE, which is phenylalanine. So A is 3. But you know that A is equal to 3 is present in all of these. Now, what about B? GGG. GGG is a codon which codes for the amino acid glycine. So that means B is for 4. That eliminates option B, which says that B is for 1. However, we need to choose still between options A and C. Let's look at UCU. UCU is a codon which is coding for the amino acid serine. So UCU is for option 1 in column 2. So the option with C given as 1 is found in option A. So that means that option A is the right option and option C is incorrect. So just to clarify, let's look at D. CCC codes for proline. As you can see, CCC codes for proline. And that's how you match codons with their respective amino acids. Let's look at the simple ones. What we could have done is, what we did first is we found out the only codon here which does not share coding with one amino acid. So this codon is only specific for methionine. So AUG methionine, E gives 2. That eliminated option D. And when you look at UCU and find out that it codes for serine, the only option among the remaining 3 that has C gives 1 is option A. So that makes option A the correct option. So that ends this episode of NEET preparation. Now, we've already made a previous episode on the chapter Molecular Basis of Inheritance. If you want to view that and other episodes that we've done on NEET Biology, then please do click on the link that we have provided in the playlist, and you can view all of those episodes. Now, if you want to access more of our useful and interesting content, then please subscribe to our channel, Brain Blitz Audios. You can get notifications regarding the same by hitting the bell icon, the notifications button, for more and the latest from our useful and interesting content, such as learner words, mind maps, memory hacks, etc. So until the next episode of NEET Preparation, take care, stay safe, bye-bye for now.